Okay, so we already talked about what is cancer, what is the statistics. You already analyzed in the last figure that age and cancer are related, and you know that is because of the accumulation of mutations. Now we're going to talk about the molecular characteristic of cancer. So I will ask you some more questions, and you will try to answer it and upload it on Moodle later. So molecular characteristic of cancer. So you will find a lot of paper that talk about different landmarks of cancer. So I'm going to show you some of them. So one of them is that they have the ability of cells be self-sufficient, so they don't respond to the normal signaling. All the things that we have been talking about, survival factors, the growth factors, all these signals, they produce their own signals, so they never died. So this is something characteristic of cancer. So normal cells don't grow unless they have certain specific proteins that are found in the environment that are called growth factor. This growth factor affect other proteins in the cell, which affect other proteins that eventually will cause them to grow and divide. So if you remember, growth factors will be outside. They go and attach to a receptor, and they will be transduced and later change in expression. Okay, this, and I know that in the previous class, if you watch all the videos, you will feel that it's like that. You know, that there is no need to memorize every single detail. If you end up study cancer, you will focus on only few of those, or if you end up doing a PhD, you will end up focusing in you will concentrate in 2 or 3 pathways, not in all of them at the same time. But right now, what we understand of cancer, I just wanted to zoom and show you that there were so many things involved, and that we are able to now understand so many of the proteins and the molecular basis of those changes. Okay, so now I want that you tell me how many pathways do you remember that are important for cancer, and I want that you write them there and tell me where is the function, what do you know about those. So you have 5 minutes to talk about those. Here there are some clues of some of those that are important. So just I just want that you start trying to remember all the information that we have discussed. Okay, so we finished with the first landmark of cancer, that they are self-sufficient, that they produce their own growth factors. The other landmark is they avoid apoptosis, yeah, so they are able to evade the signal that say died while they produce their signal that say survive. Okay, so I'm going to give you more information about apoptosis. So we talked that apoptosis is programmed cell death. I'm sure that you in the pathways you should have talked about apoptosis. Maybe if you remember Esri talking, and she said, you know, JNK pathway is important. And here you have these 2 molecules that are very important. Yeah, proteins that are pro-apoptotic and anti-apoptotic. Okay, so when you have a lot of proteins that are show you here, when you have a lot of proteins that are pro-apoptotic, so apoptosis will happen. So these proteins are telling are sending a signal that it's time for the cell to die. So you have more apoptotic signals, like for example these bags, very important. And so there is no survival cell, the survival signal reduced, while the apoptosis, the signals here, they increases and the cell died. And however, the opposite things can happen. Yeah, so show you here you have the opposite case. Now the survival factors are the one that win. So you have more of them. So the anti-apoptotic are induced, and here you have this, for example, MCL3, that is anti-apoptotic factor, and these are proteins that are telling to the cell, you know, survive, you don't need to kill yourself, continue here, you're in a good shape, you don't have a lot of mutations. Yeah, so to summarize, you have these 2 type of proteins that are depending if there is more of the other one, or if there is pro-apoptosis or anti-apoptotic, it will decide if the cell needs to survive or the cell needs to die. And of course, what happens when there is damage, they will be more pro-apoptotic. The problem is when cancer appear, and although if cells have damage, they will not go to apoptosis. Okay, so you already saw this movie in the beginning of the class, yeah? That I told you so, and we saw everything that is happened. So I want that you try to tell me what are the difference. Super easy, yeah? So write it down, put it on Moodle. Okay. So, so now we are going to talk about how cells avoid apoptosis. So cancer cells don't die easily, therefore they have more anti-apoptotic proteins. Yeah, so as we saw in the previous case, this will be the case of cancer cells that, independent of what is happening in the cell, how much damage there are more survival signals. And I'm going to show you a video that summarizes all these topics.
Apoptosis, or programmed cell death, is one of the mechanisms by which organisms limit the growth and replication of cells. If apoptosis did not occur, there would be no way to control cell growth and tissue homeostasis would be lost. In fact, this is one Apoptosis, or programmed cell death, is one of the mechanisms by which organisms limit the growth and replication of cells. If apoptosis did not occur, there would be no way to control cell growth and tissue homeostasis would be lost. In fact, this is one of the key mechanisms behind cancer. The genetic alterations in the cancer cell not only lead to increased cellular proliferation and growth, they also lead to loss of apoptosis. Along with too much cell growth, there is too little cell death in malignant tissue. Apoptosis occurs in normal cells to allow for removal of damaged cells, maintain a constant number of cells in regenerating tissues, and is an important part of embryogenesis. In an average human adult, 50 to 70 billion cells undergo apoptosis per day. Apoptosis is characterized by changes such as cell shrinkage, mitochondrial cytochrome C release, fragmentation of cell DNA into multiples of 180 base pairs, and the ultimate breakage of cells into small apoptotic bodies, which will be cleared through phagocytosis. Phagocytosis is a process where cells take in cell fragments or microorganisms in membrane-bound vesicles. The vesicles fuse with lysosome-containing proteases, and the engulfed material is processed for recycling. There are two pathways that can activate apoptosis. The first is the death receptor, or extrinsic pathway. It is triggered by activation of members of the tumor necrosis factor receptor superfamily. The second means of initiating apoptosis is through the mitochondrial or intrinsic pathway. This is set in motion by DNA damage. Both pathways ultimately stimulate a set of enzymes called caspases. The caspases interact with inhibitors of apoptosis proteins, or IAP, and the BCL2 family of proteins, which individually have either pro- and anti-apoptotic properties. In some malignant cells, there is resistance to apoptosis due to overexpression of anti-apoptotic proteins. For example, survivin is an IAP that is found in many cancers and predicts for poor outcomes. And BCL2 is overexpressed in B-cell lymphomas as a result of the translocation of its gene. Conversely, deactivating mutations of a pro-apoptotic molecule like BAX is seen in some gastrointestinal tumors and leukemias. Anti-cancer agents have been developed targeting anti-apoptotic molecules. For instance, short segments of DNA complementary to the RNA of BCL2 or antisense oligonucleotides have been designed to reduce the translation of this anti-apoptosis protein. Activation of transcription factors can lead to apoptotic resistance. This occurs, for example, when members of the nuclear factor kappa B or NF-kappa B family of transcription factors are overexpressed in certain tumors, which lead to increased transcription of anti-apoptotic members of the IAP and BCL2 families. The ubiquitin proteasome pathway regulates the expression of transcription factors and other cell cycle proteins. Certain molecules can suppress or reduce NF-kappa B
so
and
this is a
AP1
pretty
active.
good video that summarizes all the information. And again, you don't have to memorize every single molecule. You will drive you insane if you think that I'm going to ask you every single molecule. But I just want that you know, very important that you know there is an extrinsic and an intrinsic pathway. I already show you 2 videos talking about that. That caspases are very important, and that there are families there are molecules that inhibit apoptosis, that are families like BCL2 that are important in this kind of process. So just that you start being aware that all these pathways exist, and then later when you're reading a paper, it's much easier for you to understand them. Okay, so let's move to the next slide. Okay, so we already talked about that they produce growth factors, yeah, proteins that affect pathways that allow them to continue surviving, that they evade other signals involving the apoptosis. Yeah, so there are intrinsic and extrinsic pathways, and caspases are very important. So remember that cancer break that signaling, and now there will be another process that is called sustained angiogenesis. So we have already talked about angiogenesis. I just remind you, angiogenesis is very important. Why? Because in the blood, yeah, if you remember the slide that we watched before, the blood transport nutrients and oxygen to the cells, so and also remove the waste products. In cancer cells, the cells grow and replicate, and they don't die. But as they have such a big cell area, then they will require blood vessels. So they need food to continue surviving. If they don't have the food, they will die. So they activate a specific factors, yeah, that allow them to blood vessels to appear. And this is, as I have said multiple times in the class, this can happen. It's a normal. It happens in normal tissues. Every time that you cut yourself or you have an injury, you need to repair some of the blood vessels. Yeah, they need to. So it's something that happens in normal tissues and are very important for tissue remodeling. However, here is put into the wrong context. So now I'm going to show you a video here. VEGF. So this is a factor that is very important that is involved in the process of angiogenesis. So again, I have been talking very general about the process of angiogenesis. However, they have found so many molecules and each one of many of the factors involved into that. So we're going to watch videos that talk about this process of how tumors can send this signal to produce blood vessels around them so they continue growing. Okay, so before watching the video, remember that angiogenesis is the growth of new blood vessels from pre-existing vessels. Cells secrete proteins that tell the blood vessels to grow and to grow nearby. And an example is this. So this vascular epithelial growth factor, yeah, so VEGF. So let's watch the video.
Tumor angiogenesis is the proliferation of a network of blood vessels that penetrates into cancerous growths, supplying nutrients and oxygen and removing waste products. Tumor angiogenesis actually starts with cancerous tumor cells releasing angiogenesis-stimulating growth factors. Tumor angiogenesis is the proliferation of a network of blood vessels that penetrates into cancerous growths, supplying nutrients and oxygen and removing waste products. Tumor angiogenesis actually starts with cancerous tumor cells releasing angiogenesis-stimulating growth factors that send signals to surrounding normal host tissue. This is the angiogenic switch, typically a result of hypoxia. Tumor blood vessels grow, and in addition, tumor cells can co-opt existing blood vessels. Several growth factors have been implicated in these processes. Looking more closely at the release of growth factors, such as vascular endothelial growth factor from the tumor cells, vascular endothelial growth factor, VEGF, is released and binds to VEGF receptors on the endothelium of blood vessels. Anti-angiogenic drugs work by literally starving the tumor of its blood supply.
Okay, so pretty good. Sorry, I made a mistake. It's vascular, not epithelial, endothelial, because it's from a blood vessel growth factor. So this, we already talked about the TFU beta, that is the transforming growth factor, or we already talked about a growth factor in the class, that in the video lecture, that it was the epithelial growth factor, and this one is the one involving in angiogenesis. And remember, here they show you one treatment that is to start, one of them hundreds of treatments, that one of them is just start the tumor by stopping the process of angiogenesis.